Welcome to Black and Microbiology's Community Day. It is my honor to introduce our guest for today, Dr. Oveta Fuller. Dr. Fuller is an associate professor of microbiology and immunology in the University of Michigan Medical School and faculty in the university's STEM initiative of the African Studies Center. She is also an adjunct professor at Payne Theological Seminary and an ordained minister. She earned her bachelor's in biology and her PhD in microbiology and immunology at the University of North Carolina, Chapel Hill, and completed her postdoctoral research in virology at the University of Chicago. As a virologist, Dr. Fuller's laboratory team has published studies on herpes simplex and influenza viruses, and she teaches medical, graduate, dental, and undergraduate students about human virus pathogens, including a study abroad course at the University of Michigan called Global Impact of Microbes Fieldwork that immerses participants in global research field sites to explore effective partnerships for advancing health. We are so happy to have her here for the Black and Micro Community Day because she is going to share with us more about the Trusted Messenger Intervention Program that she developed to engage networks of religious leaders and communities of Zambia, South Africa, and sites in the US that enable them to effectively engage with their community in infectious disease prevention. During the 2020 global emergence of SARS coronavirus 2, into the COVID-19 pandemic, Dr. Fuller has engaged widely in transfer of science-based insights to community through conversations, webinars, articles, visual and other media to networks of religious leaders, seminarians, university students, African-American community leaders, and to companies and the public. She serves on the FDA VRBPAC Vaccine Advisory Committee. So basically, she's all that, and we are so blessed to have her here with us today. Dr. Fuller, I am so happy to know you and to be able to moderate this session.
It's a pleasure to be here. I'm so grateful for the invitation and to be part of the second Black in Microbiology Week. So exciting. You guys do such a great job.
Thank you. So you're going to share some slides with us today, right?
Yes, I was told that this is Community Day, and there are days that have been focused on research and STEM. And so what I wanted to do was to take us on a little bit of a historical journey and tie that with the work that I've done previously with HIV and now things that are happening. Hopefully that will be informative also for people about COVID. And so you can stop, and there's going to be a place where we'll be asking questions, or there'll be a chance to ask questions. So we'll make this a little bit more conversational perhaps than your typical talk, if that's okay.
Sounds good.
All right, then let's do it. So this again is Community Day of the second annual Black in Microbiology Week. And so when I was asked to share a little bit, the title that I've been using recently was modified a bit, but so we're going to use the title From Milkmaids to Messenger RNA, Equipping Trusted Messengers. And I don't know about you, but I used to read Mother Goose Tales as a kid, and there was always something about the fair milkmaiden in there. So we're going to talk about that a little bit in this discussion. So one thing that stands out for me as a scientist and as a person is that this quote, which I think means a lot if you think about it. Science is but a perversion of itself unless it has as its ultimate goal the betterment of humanity. And this is a Nikola Tesla quote. And the question that I ask, and many people ask, as a basic scientist trained person and someone who studied herpes simplex for years and who's fascinated by viruses, is how do basic molecular biomedical science and clinical research, all that we do in the laboratories and in field studies, make a positive difference in the everyday life of people? So how do we get the science off the shelves and the publications into the real life? And that brings the question, can science and religion work together as effective partners in issues of health? So the answer, of course, because we're here at Community Day, we're scientists, many of us are African Americans or Latinx or some other ethnicity, First Nations, is that we want to do science that affects our community because we're from our community, we're in our community, we know there are needs in our community, And we know those needs don't always get met by the larger society. So we feel sort of a heavy responsibility that with our education and opportunity is the need to return to the community and give back and make it better for those who we represent. And so what does that require? That is possible through and is necessary, not just for people of color, but for everyone, as we can see in the current pandemic. Effective, mutually beneficial community university government partnership is required. And so this is just a picture from some of the work in Zambia with Trusted Messenger. These are students from U of M who are working with me in the workshops for clergy and religious leaders. And then these are some of the community sites. This is a school that was open for vulnerable children who had lost a parent to HIV/AIDS. And this is a minister who had just gotten a license to keep his school open, even though it grew bigger and outgrew their bathroom capacity. They had a donor that helped them build a bigger bathroom for all the students that were coming. And this is his license. And so he's so happy to be able to show that. But this all happened as a result of much cooperation between all of these entities. So what I want to do today is take you on a little journey about pandemics and look at 3 viral pandemics. This is a history of pandemics all the way from the plague back in the 1st century to the current time of COVID-19. And so we're going to stop on the way looking at smallpox, which about over 56 million people, as we can tell, died from. HIV/AIDS, which is what the Trusted Messenger intervention that I do cut its teeth on, and then the COVID-19 pandemic. And so pandemics have in common that they are prevalent all over a country or a world. And we see that here's an early 12th century picture where a king had smallpox and the clergy have been called in to pray over him. So we see that the community has always been involved with infectious disease as captured in art. And then this is kind of a deja vu thing too. These are pictures of protection that was used back in the 16th century with plague doctors. And it just reminds me so much of what's happening now with COVID-19. Here's someone doing a COVID-19 test. And it's like, you know, the more things change, the more they stay the same, because we still have the same sort of covering that was looked at years ago to protect those working in the community. So what are trusted messengers? I am not the first one to use this term. It's a well-known term in social areas, where they are credible sources of people who can influence community. So religious leaders, for example, are a credible resource in reframing community perceptions. And in my case, about disease control and prevention. And not only me, there are lots of cases in the literature where people have utilized religious leaders or the faith community. It's just that we try to do it in a systematic science way to work with networks. So the strategy of the trusted messenger idea we have for working with infectious agents on a systematic way is to use in-depth science-focused education with established networks of religious leaders. So those leaders become competent and effective at addressing whatever it is. In the first case, it's HIV/AIDS, which of course had a stigma around it and has so many places where faith leaders could fall in potholes if they dare to talk about this. Or COVID-19, we see now what's happening with the mask dilemma and the vaccine versus not vaccine, or whatever is next in the world of infectious disease epidemics and pandemics. We need 2 types of trusted messengers. One are knowledge informers, like scientists like myself and physicians and others who know the information about whatever it is that we want to change. But then there's another kind of trusted messenger called the value informer. And these are people who know the values of the community that they're working in. So that's why I say many of us in science who are people of color know the values of our community. We know how to speak the language. We know the things that are important. We know the traditions. And that gives us buy-in and credibility with people in our community. So this is a wonderful thing to remember. A message is only as effective as the person delivering it. And to be effective, one needs the right message that's both right in knowledge and value. It needs someone who either is a value informer that the community trusts or a knowledge informer that they trust. And then it needs a means of distribution. And we can only look at the COVID pandemic to see where we've gone wrong in many of these aspects. Oh, and by the way, this was my first team of students that worked in the field in Zambia in 2010. Many of these have gone on now to finish grad school or professional school or physicians. And then we had partners that were part of the community that traveled with us. So it was a partnership between the AME Church in Zambia and the University of Michigan Microbiology Emergent Team. All right, so let's start this journey with these three pathogens that we're talking about. We all know that we're in a coronavirus COVID-19 pandemic, and probably most of the people here are aware that this is a naturally emerged zoonotic virus, meaning that it was an animal virus that moved into humans. And we know there's 7 different coronaviruses that have become human viruses, of which we are not always thinking about these, but there are 4 different human respiratory viruses. And then there's SARS, which was an outbreak in China in the early 2000s, and MERS, which is still even now in Saudi Arabia, Middle Eastern Respiratory Syndrome. And then there's COVID-19, which was declared a pandemic in March of 2020, as we all so very well know. So SARS coronavirus 2, that's a new human virus, new to humans, has moved around the world as a zoonotic adaptation to the point that there have been over 232 million cases documented, meaning lots undocumented, and over 4.7 million deaths. That clearly affects every community, almost every community on the planet. So what are these things that we're dealing with? Emerging microbes and epidemics and pandemics will continue to occur. Emerging diseases are when something is caused by a microbe that first appears or increases in incidence in an area or new location. So clearly COVID-19 was emerging out of Wuhan or wherever it started to now become a worldwide pandemic. And really it happened in less than 8 months. An epidemic is where something is affecting a large number of people in a location. A pandemic is when you have multiple epidemics. So it could be an entire country or region, or in this case of the 3 viruses we're talking about, the entire world. And then we can have resurging microbes such as measles when we don't take the controls necessary to keep the microbe at bay. There are all kinds of factors that affect the emergence of microbes, from the enhanced animal to human interaction, to mass distribution of food and travel, to genetic recombination, like with the COVID variants, to changes in human behavior and things like population growth or housing or war and civil unrest. So we will have more epidemics and probably some more pandemics. So the one of the microbes I'd like to share about is smallpox from a historical perspective. And it has a significant place in the history of infectious disease and science. We know that Ramses V had smallpox based on the mummy that has been found, and we see the pox lesions as shown in this picture, and we can see those scars on his face. As a matter of fact, milkmaids, which we read about as the fair milkmaiden in the Mother Goose tales, they were fair not because the color of their skin, but because their skin was not scarred by the remnants of smallpox. And why was that? This slide shows you, this is a woman who not only had smallpox, but who's blinded because of smallpox. It has played a major role throughout history, with over 500 million people being killed because of smallpox just in the 20th century. We know that it's been used as a biological warfare. So Cortez brought blankets that were formites for people who have been around people with smallpox, and used those to help weaken the Aztec Indians as they overtook that area. So it's been a major issue in the world. And yet science and the scientific method was used to help us eradicate smallpox. And it became the first example of a proven vaccination. So the story involves Milkmaids, which is part of the title of this talk. Edward Jenner, who was variolated, meaning he was given a little bit of material from a smallpox lesion and almost died, did an experiment where he noticed that milkmaids who have this fair complexion did get cowpox, but they never got smallpox. And so Jenner took a little bit of material from a cowpox lesion and inoculated an 8-year-old James Phillips with that material. And then about 6 weeks later, he challenged him with smallpox, with the real deal. And the young man did not get infected. So Jenner said that the exposure to the cowpox protected him against smallpox, which is true. And that process then was the first scientific proof of protective immunity or vaccination. Vaca or vaca comes from cow in Latin. And so that's our first successful example of immunization from the observation about milkmaids. This, of course, there are many artists' versions of things that happen. And Jenner had a really hard time publishing his paper, by the way. People predicted that folks who got this vaccine would grow horns and all kinds of crazy conspiracy and misconceptions. And Chelsea, if you're still here, Dr. Spreeks, does that sound familiar?
It sounds very familiar.
So here's a picture of the cowpox being extracted and then used to inoculate people to protect them against smallpox. Because remember, lots of people died. It was a rite of passage as to whether your child could get through smallpox or not. Somewhere along the way, we stopped using smallpox or stopped using that cowpox and moved to a variant vaccinia virus as the vaccine. And so between the 1800s and the late 1970s, with a major campaign and technology of heat staple and freeze drying, we were able to administer the smallpox vaccine around the world and use public health isolation approaches to be able to actually eliminate smallpox, to eradicate it. And there's some reasons this was true about the virus that I won't have time to go into, but the great part is that we no longer have smallpox. I have actually a smallpox vaccine, and so you can date people based on whether they have this little lesion on their arm or the scar, which is from the smallpox vaccine. We stopped doing that at one point, so anyone who has that has to be over a certain age. And you can see how this disease was very devastating. But thanks be to science and technology, smallpox is no longer an issue in our lives. But we do have others. And one of those others is COVID-19. This is the current graph of the USA levels of cases of COVID-19, where we had the first peak that we thought was wow, and then we had the second peak that was even higher, and then this third peak or surge in from around Halloween through the middle of February, where so many people were sick and so many people died, is the big one so far. We're looking now at, as we enter back into school, another uptick of this, another surge, and the latest data show that it's going down, hopefully, but we'll have to wait and see if that holds up. The deaths typically follow behind the number of cases and hospitalizations. Unfortunately, since about the last of December or the first of the year, we now have vaccines that are highly effective against COVID death and disease. And so right now in the US, we have about 55% of the country fully vaccinated, and that includes those down to 12. So we still have a long way to go, but we are making progress in trying to keep this down through a vaccination. So what are these vaccinations? Oh, I forgot I put this in here. This slide shows you that the illness that coronavirus caused in the COVID-19 pandemic, as many infectious diseases and many other chronic diseases, is disproportionately present in communities of color in the US. And there are a whole lots of reasons for this, many social behavioral issues, as well as the deliberate systems of inequality and racism that also was pandemic revealed this year. This shows you the US level of coronavirus as of October last year, showing that we are leaders in the world in the total number of COVID-19 deaths and cases. And imagine that, a very developed country like the USA. This shows the cases in Chicago and the deaths in Chicago among Latinx, Black, white, Asian, or others. And look at the death rate among the deaths from COVID that were among African Americans. This gives you the data in another way. So if we look at our state, Michigan, this axis gives you the population percentage that are African Americans, and this the number of deaths as the record showed at that time in a particular place that were of African Americans. So we were 14% of the population, but 41% of the deaths recorded by May 2020 were African Americans. So this is disproportionately affecting communities of color. And of course, scientists who are part of the community of color are really tuned into this sort of thing and want to make a difference. So we also know that we're very fortunate to have scientists who have developed with cooperation of government and one another COVID vaccines. So what about those COVID vaccines? As Dr. Spriggs mentioned, I'm fortunate or serving on the FDA panel that is advisory about vaccines, and this is one that came up. So we have 3 now approved vaccines in the US, emergency use approved vaccines for use against COVID-19. 2 are messenger RNA vaccines, and 1 is a vector carrier. They're all, and these were all being studied around the world in phase 3 trials. They're all looked at for efficacy and safety. These links are great resources to tell you the technology behind messenger RNA vaccines that I won't go into today. They all have gone through these, all 3 have gone through the clinical phase trials that are used, the system that is used to make sure that drugs and vaccines are safe as well as effective, and including a large clinical trial that was reduced in its time, but increased in the number of people to get the same power of effectiveness. So the Pfizer vaccine, which is now licensed, recently licensed, and the Moderna vaccine, as well as the Johnson & Johnson, had thousands, tens of thousands of people in their phase 3 clinical trial. And they're all still now being looked at in terms of long-term surveillance to see what happens with people who, how long the vaccine protection lasts or any other effects that it has. So these have gone through the necessary rigorous studies. So what is in those messenger RNA vaccines? This is a diagram of coronavirus or COVID-19 virus or SARS coronavirus 2 with an RNA genome, and one of those genes is for the spike protein, which is in red here. That gene has been made into a messenger RNA copy or code and placed within lipid nanoparticles such that it's only the messenger RNA, the code for the spike protein, in a fatty acid or fat lipid droplet. That then is suspended into water and sucrose, and that's what gets injected into our arms when we get a vaccine, a messenger RNA vaccine. The neat thing about this vaccine is that it allows the lipid that is producing, that has the messenger RNA, to enter into cells, immune cells and other cells, where our own cells make the spike protein to be expressed on cells, recognized as a foreign antigen by antigen presenting cells, and then stimulating immune response just to the spike, not the whole virus, just to spike epitopes, such that we end up with a memory of B cells and T cells and activated B cells and T cells that make antibodies and memory cells. The point is that when we see the whole virus, because we are now recognizing the spike, we will be able to prevent the virus from binding to cells and causing disease. So memory is established by putting in the little code, not the virus. The code decays in a few hours, but the protein is expressed long enough so that immune system can mount a response to it. We actually, in the study, the studies by the pharmaceutical agents looked at the side effects, which last only a few days, and they're pretty similar with both of these. And we know that both of the mRNA vaccines are highly effective against disease and death, even though they're slightly different in some of their storage features. Pfizer is authorized to be taken down to age 12, whereas Moderna is age 18, and there's slight difference in the time between doses. But essentially, they are amazing feats of science to help us have another tool for blocking coronavirus disease and deaths as we fight this pandemic. And so in summary, what do we need to deal with the COVID-19 pandemic? I don't think we're going to be able to eradicate coronavirus like we did with smallpox, because there are features about coronavirus, like with smallpox, there's really only one genetic variant of smallpox, and also there are no animal reservoirs, and those are necessary features if you're going to be able to eradicate a pathogen. And also immunity gives you, infection gives you lifelong immunity. Those things are not true with COVID. But in order to get to a place where we can coexist and manage COVID, we're going to need all these things. We're going to need trust from people to do what's necessary to stop the virus from reproducing. We need transparency from scientists and physicians about the pharmaceutical companies about what's going on, and if there's a problem, stopping and examining that to figure out how to fix it or how to get around it. We need to include diverse populations. Not everything works the same way in everyone. So fortunately, the clinical trials had quite a bit of variety of people. We need equitable access. We know that getting access to tests and to vaccines early on was not so easy, particularly for people of color, just like getting to a doctor's appointment in some areas is not easy. We need community engagement. We're talking about community today. So we need community organizations and churches and sororities and frats and other clubs and families to engage. All of this to build trust. And we also need clear messages. I dare say we haven't had the best and most clear messages from the beginning of the COVID-19 pandemic, but I think there are efforts to try to fix that as much as we can, even though it's all very fluid. We're in a changing situation. And then we need trusted messengers, people who can take these clear messages and communicate it in a transparent way to the community so there's high buy-in to use the tools we have to protect the population. So whenever I talk about COVID vaccines, I also have to bring in that the disease itself, we don't know a lot about it, and yet it is a very devastating disease. It is an aerosolized virus, so easy to transmit from person to person when we talk or sing or breathe or dance. Anyone who's infected will be putting virus into the air that can be breathed in by anybody who's close by. It replicates in the nasal passages, goes into the trachea and tissue of the lungs, where a healthy lung tissue in the alveoli can exchange air and CO2 through these air sacs. A coronavirus infected lung, due to the virus replication and the immune response, ends up with swollen mucus clogged tissue that no longer can exchange the oxygen and CO2, ends up with fluid in the lungs, and thereby affects other organs. And that's what's known as severe acute respiratory syndrome. We know this happens, and we know the vaccines protect against this. So this is a no-brainer in my mind, that if you can get something that's been through rigorous safety and efficacy studies to keep this from happening, that it's a good idea to do. And even if you don't get sick from COVID, there are people who can express virus or make virus and they never get sick, but they can cause somebody else to be infected. Not only does COVID-19 affect the lungs, but the lack of oxygen to the blood and to the body affects other tissues. So heart arrhythmias, heart attacks, blood clots, kidney damage, immune system damage, blood clots, brain issues, even things as mild as pink eye and loss of taste and smell. All of these are potential fallouts of COVID-19. And so when somebody has severe COVID, the goal is to try to keep oxygen in their body, and that is going to depend on whether they live or die. Even people who get mild COVID or asymptomatic infection can have what's called the long hauler syndrome, which means that they may or may not be producing virus in a test, but they just don't feel well. People who have been athletes or people who've been in great physical shape end up not having trouble walking up the stairs, or brain fog or vertigo or chest pains or insomnia for months. And we don't understand how this happened. So there are many questions unanswered about COVID-19 disease itself. And yet the vaccines that have been produced amazingly by scientists and others cooperating seem to block the more severe disease. So in summary about COVID and vaccines, the messenger RNA vaccines, we have 3 vaccines, 2 messenger RNA and 1 vector, an adenovector virus, a virus vector vaccine. These are the symptoms of COVID, which people get some and not all, and some may get no symptoms at all. But the vaccines prevent against severe disease. And so COVID vaccines reduce disease symptoms, illness, and death. And recent evidence shows, however, there's still virus shedding that goes on even in vaccinated people. So everybody, with or without a vaccine needs to use all the tools we have, which include masking, distancing, not gathering, or being careful who you gather with, so that we can stop the reproduction of COVID-19 and the variants. So we're in this thing together. We're in a historical time of a pandemic. COVID-19 has taken the world by storm, and our communities, every community needs people who they can get accurate information, both knowledge informers and value informers, so that they can integrate and use the things that we are learning. And there's a lot we still don't know. So I'm going to stop here for a minute. The rest of the talk is just simply to tell you a little bit about Trusted Messenger with HIV, which is where it was developed. But I'm going to stop here and see if there are any questions or comments. The question I have is, how do we all together move to coexist with COVID-19 and SARS coronavirus 2? Will we be in this pandemic another 6 months or another 2 years or another 5 years? Or will we just learn to coexist? Or will nature do something and the virus will magically go down? I don't know. So I'm going to stop sharing the slides for a minute and see if there are any questions or conversation about that from our host.
Thank you, Dr. Fuller. That was awesome. I really appreciate you taking us through the history of vaccines and especially how this COVID-19 vaccine works. And so it seems like the population is kind of split. You either want the vaccine yesterday, or there's no convincing people at this point to get it. And for some people, it is political, but for a lot of people, especially in our community, it's more them just having questions about whether they can actually trust the doctors and the scientists in the hospitals based off of, history. And in the center of that diagram you had was building trust. So how do we as scientists and microbiologists, or people who understand the science of the vaccine at least, how do we build trust with our friends and family members and colleagues who just do not know whether or not they can believe the science?
Yeah, so that is the challenge, because there are many people who are not avid anti-vaxxers. They're just not sure enough to take this step. And so I think people who understand this really can make a big difference just by talking to people, just by, asking your auntie or your grandmother or your cousin or the kid down the street, have you got, particularly teenagers and young adults, have you gotten your vaccine or have you gotten the vaccine? And they can either say yes or no. And then, sometimes people say, well, I'm not getting that, and you can ask, well, why? And then share, well, you didn't ask me, but would you mind if I tell you what I think? And just tell them, this has gone through rigorous trials, or they'll usually say, oh, I'm not sure that was studied well enough, or, I've heard that it makes women infertile, or, all kinds, or I hear that it will change your genetics. And there's so many misconceptions that are circulating on public media and other places. I think just having the conversation with people, and, sharing, like when I wanted to wait for a few weeks to see the big rollout and to see what happened before I got vaccinated. And I finally said, okay, I'm either going to do this because I don't like putting anything in my body that I don't know, believe it or not, what it is. I mean, I do that all the time, but I don't like doing it. So my husband says, well, I think you're just going to have to step out on faith and realize that getting protected from COVID is really important to do. And I thought, that really does make sense. So in mid-January, I went and got the vaccine, right? And so I think sometimes you just have to discuss it with people. You just have to find out where they are and share in love with them that none of us really understand COVID-19, but we know that it's a very serious issue And that if something can help us, including masking and distancing and not getting together, you know, in unresponsible ways, that we should do that.
Thank you. Yeah. Along those same lines, you know, we talked about how there's pandemics and epidemics every so often all throughout history. And so given the repetitive nature of these pandemics, how can scientists better engage with community leaders now to make sure that we're prepared for the next pandemic instead of trying to play catch up like we've basically been doing with COVID-19?
I hope that COVID-19 is teaching us that preparedness is so essential. And not only preparedness in terms of emergency response, but preparedness in training people in cross-disciplinary ways, and having value systems that say, yes, doing basic science is absolutely essential and critical, but doing public health science is absolutely critical. And then having a few people who can navigate both of those is absolutely critical. And what I can do if there's time is to share with you a little bit with the trusted messenger process that is a way, other than personally talking to people, a way of working systems and networks to maybe have a larger impact on a greater number of people, or better yet, to develop trusted messengers that can now activate their own community. So should we continue with that part?
Yep, let's keep going.
All right. So let's see, let me get back to my share screen. All righty. So, I forgot I put this in here. So for the COVID-19 pandemic, since March of 2020, or actually February 2020, when a colleague in the AME Church, Dr. Miriam Burnett, who's head of the International Health Commission, and I realized that this is an aerosolized virus. It's in China now, and we think we can keep it out. No, this is coming. This is going to happen. So these are some of the many, many ways that we've been, and others who are considered trusted messengers, or people who have the knowledge base, have been asked to engage with community. This is with the Bishop Hezekiah Walker Fellowships, which is a global fellowship. This is with a group in Maryland. This was a wonderful talk at the Fralin Biomedical Research Institute. That was a community talk in Virginia. So essentially the opportunities have come such that I'm not always sure whether I'm talking about COVID with Trusted Messenger, or I'm talking about our HIV research that we use to actually use this method. So I'm going to tell you about the HIV research now, and hopefully I won't say COVID when I mean HIV. This is one of our teams of students that work with me as research assistants at the Tropical Disease Research Center in Ndola, Zambia. It used to be part of the World Health Organization, but now it's an independent research center. So these are students at Michigan who were either pre-med or pre-grad or pre-public health, working on a research field project with HIV/AIDS and Trusted Messenger. You'll see some of the data that Taiwei analyzed a little bit later. And so what we were initially, Trusted Messenger, again, which is a concept in sociology, but we were invited because, as you mentioned, I'm a minister, and was looking to do sabbatical in 2005 and wanted to work with our Service and Development Agency with HIV/AIDS. Well, the bishop from Zambia realized that HIV/AIDS was devastating his community, such that people were, ministers were doing 3 funerals a day. I mean, more than they could handle, because this was a worldwide pandemic that we had recognized from the late 80s when it became, or actually when the virus was first discovered. And if you'll notice with this figure that over 75% of the cases of HIV, particularly back in the early 2000s, was in sub-Saharan Africa. And these are my folk, right? These are my people, right? So even now, 2019, the total number of people living with HIV in the world is over 38 million. And, you know, it's like this epidemic or this pandemic has not gone away. And so I was invited to work with the people in Zambia to help the clergy understand what HIV is. And the AIDS pandemic took a major change in 2011 when the pre-treatment using antivirals was shown to block the transfer of HIV from an infected spouse to the uninfected spouse. So if both people are on antiretroviral drugs, the virus does not transmit, which means that we could stop the transmission pattern of this virus. And so that would brought up lots of magazine covers like this, The End of AIDS. And there's campaign, of course, in the US ending the HIV epidemic by reducing the number of people that are infected by 90% in 10 years. So the point is, the thought is we can get to the end of AIDS, but how do we do that? Just like we got to the end of smallpox through a very purposeful vaccination campaign and natural immunity. So what we have to do for HIV is use the wide range of tools in our toolkit, from screening mothers, pregnant mothers, to make sure they're not HIV positive, to the old-fashioned way of using condoms, to treatment as prevention, that is given the antiretroviral drugs, or PrEP is one of those, to integrase inhibitors, and all these things that have come out of basic science discoveries about how HIV works. And the goal then is to know whether a person is positive or negative, meaning we all need to get an HIV test. If you're negative, stay negative by avoiding the virus, and I'll show you how we share about that. Or if you're positive, you want to get linked to medical care, get assessed as to where you are in the HIV to AIDS process, get into care with antivirals, stay on those antivirals until viral suppression is reached. If we could do this, we could actually get rid of HIV. And so that's where trusted messengers coming in. Trusted messengers, remember, are people who are credible resources in the community. So we propose that clergy and religious leaders' perspectives can be changed so they reframe HIV/AIDS or COVID-19 as a preventable disease. So this is an individual level intervention as opposed to a population level. We're looking at what happens with individuals when we help them to get a different perspective or reframe HIV as a preventable disease. This intervention engages faith leaders as gateways to mobilize available resources. And we're not just doing just any faith leaders, we're doing religious leader networks. So it also uses rigorous qualitative and quantitative documentation. After all, I'm a scientist, right? So we need to document, we need to have the data, the evidence, the scientific proof that what you're doing makes a difference. And we do this in networks as opposed to working with individual leaders. This came about, as I mentioned, by an invitation in 2006 to work with the bishop in Zambia, where the population had about 13% HIV prevalence. Indeed, a very difficult situation. This is Zambia. It's about the third country north of South Africa, a butterfly-shaped country. And this shows you that the places of the 9 provinces, the overlap of where we have high, in my case, African Methodist Episcopal Church membership is also where we have high HIV. I almost said high COVID-19, but high HIV. And so what we decided to do was to use the bishop's authority to get people together so they would come to listen, and then we would give them the science information that would help them understand HIV as a preventable virus. At that time, only about 30% or less of the people had been tested, even though HIV testing was free. Whereas now that has changed, and that a large number of people, due to lots of things with the government and Ministry of Health and NGOs, have moved that needle up to about 67% of the people tested for HIV infection. So what did we do? What did we do with Trusted Messenger? So it's not just giving them information, it's a mixed methods, intentional approach of engaging community through, first of all, focus groups to find out, why do you think HIV is affecting your community? It may be different in the mining region of Copperbelt than from the tourist region of Victoria Falls and Livingston. So we meet with the highest leaders that call themselves together because they want to do something about the issue in their community. So it's not like we choose the community, they choose us. Then we design a time to come back where they announce it to their networks. They provide the room. We just bring ourselves and things we need, and usually some funds to make lunch or have lunch made at the site. We don't give transportation reimbursements, and this is their buy-in. If they want to get this, they have to have some skin in the game, so they get there. And so it's a 2-day workshop. The first part of the day is getting the surveys done, because we want to do the baseline level of knowledge and what their attitude is, and have they been tested, and what are their actions at baseline. And then we do a PowerPoint about the biology of HIV, which I'm going to show you a little bit of in a bit, just to give them the understanding that this is a fragile virus with a lipid membrane that cannot dry out, or the virus becomes inactive, and it can only transmit in a few ways. Day 2, they go home, and day 2, they come back. There's questions and answers and discussion about misconceptions. And then they get together in small groups to strategize about using what they know about their communities and what they've learned about the science of HIV/AIDS to set up ways that they can make a difference. And then at the end, there's this huge celebration about now that the light bulb has gone on for them, or so they say. And then within 3 to 6 months, we come back to do a follow-up survey to see, what have you done? Did you get tested? Did you change your leadership? And we also visit sites to see what they're doing, as you'll see. We have short-term goals of increasing the testing level, sustained HIV accurate information, mobilizing leadership to use resources that were already in the community but unused, and then decreasing stigma and paralysis. And then there are long-term outcomes like the effects on infection and AIDS, which we don't measure, but which are measured by the Ministry of Health in the regions where we've worked. So that's the setup, with the idea that you have, whatever you do, you need to know what you're doing, tweak it to make it work, and document whether it's working or not. That's what science is. So I'll take you now very quickly through some slides that show you actual items from the workshop. This is one of the first groups that we did. This is a group of young adults who were sent by their bishop representing the church to learn about HIV/AIDS, and then become people who could go back and encourage others or model behaviors of testing. So this is the first slide in one of the workshops, day 1, what effective clergy should know. We're going to talk about what is a virus, how does HIV virus lead to AIDS, and how do you stop this? With the take-home message being that this is preventable. We can stop this thing. It's not magic. It's not God's punishment. It's none of those things. It's just a tiny, fragile virus. And so we talk about the virus and the lipid membrane, and that it can only be transmitted by blood, semen, vaginal fluid, and breast milk. So if you don't come in contact with any of those, you'll never get HIV. And that's absolutely true. Of course, this means that you never have sex, but there are ways to get around that, because we have things like condoms and other ways to block contact with semen and vaginal fluid. We also talk about the life cycle of HIV, which I'll come back to. We talk about the origins. Where did this thing come from? No, it wasn't made by the USA or other developed countries to take land in countries of Africa or other developing countries. We talk about the timeline from the first infection through people getting really sick with AIDS, and how with the antiretroviral drugs that are available, the ARTs, we try to keep people with a high T cell level, which is the helper cell levels that allow us to fight off things like influenza or herpes or pneumonia. And we want to keep the viral load low. So we actually go through this kind of thing. We talk about the symptoms which they have seen in people that they love or family members. And we talk about how to prevent HIV infection. So blood banks are screened, so it's not blood transfusion. Prenatal care for mothers and how important that is. If they're positive, they're put on antiretroviral drugs so the child doesn't get infected. What individuals can do, how we can stop the spread of HIV, how getting tested is such a critical part of regular healthcare, and that you don't have to worry about being stigmatized about getting tested if everybody's getting tested, including the pastors and the missionaries and the adolescents and everybody. Then these are the ABCs: abstaining from sex or IV drug use, which is possible, being faithful to one person who's negative and you're negative, or if you're not going to do A or B, consistent use of condoms. The point is, you can do something to prevent HIV transmission, and getting tested is the critical first step. We talk about what the test is. We talk about antiretroviral drugs where they interfere with the life cycle of HIV. And we really talk about this. We demonstrate this. This is not watered down for the faith leaders. This is a picture of the mechanism of AZT, how it stops the elongation of the genetic material of HIV. And so that's what we do in the PowerPoint. And then they have the surveys before and after. This is a group in Ndola taking one of their surveys. You can see me at the front of the room and one of our research assistants here. And so looking at some of the data very quickly, this is quantitative data from early on where we gave a 20 question survey about what do you know about HIV and how it reproduces. The clergy and pastors scored the lowest coming into the room, whereas the young adults were much higher. But going out of the room at the end of the 2 days, everybody had increased. And look what happened with these pastoral leaders. I mean, look at that. They were just sponges soaking up this stuff. And then the proof of the pudding is when we went back for the follow-up 12 months later, we asked people, go get tested if you understand how important that is. So when we went back without any prompting or any review and did the knowledge survey, a version of it, the people who returned scored at 85%, remembering the material they learned. But the thing that was most incredible is that that 71% of these returning people, granted these people returned, so they might have been a biased group, but 71% of them have been tested in that year compared to a typical 30% of the population. So we were really excited about this data. But you know, in science, you have to ask questions. Well, did this happen because of what you did, or was it media in the area, or was it some other public relations campaign that the Ministry of Health did? How do you know that the results you see in a population or in individuals are due to what you're doing? So that led me to apply for a Fulbright, which was awarded. And so I spent 10 months in Zambia doing what we call the Copperbelt Crossover Validity Study, where we worked with religious leader networks in a particular region, having half of them in a study group and half of them in a control group, and then doing a crossover such that the control group also got the intervention. And so what we found is that the results of getting tested or changing what you do followed only after they got the workshop. And so here's an example, a pre-test mean of 74% correct, post-test of 84% on understanding what HIV is. And these are other data that I won't take time to go through, but showing that the testing increases, and it increases after we do the workshop, not before. So it means something in the workshop was a stimulus for people to go get tested. Same is true with leadership. 28% of the pastors never talked about HIV, whereas after the intervention, that was less than 3%. So these kind of quantitative data tell us that there are changes that are made by the religious leaders once they get the science valued information from people they trust. They go and do things differently. And then there's qualitative results as well. These are pictures from various workshops. And so this is a study that's published now that was done in collaboration with a colleague at the School of Public Health and a graduate student, where he did 1-hour interviews with participants from these various workshops with these kinds of demographics. And so he was asking them, what did you get out of the Trusted Messenger workshop? And so they're saying it was comprehensive. It was simple but profound. It was enlightening. It was eye-opening. And so this paper is full of some of the quotes of those people, and I'll just give you a couple. So for people living with AIDS, knowledge acquired is transformative. Hilda, who's HIV positive, said, I didn't know deeper what was happening in my body. Trusted Messenger showed me exactly how to use my mind to understand my body. She said, it's important because you can control it. You have the power over HIV/AIDS. You know what to do. Maybe if I'd known this information earlier, my CD4 count would have been higher. Or this is a clergy person in her 40s who talked about how that helped her to be a better clergy person. When somebody has got the knowledge, you can tell them this is not a death sentence. There have been people that have lived so many years living positively. So then they will accept their status, and she's meaning as HIV positive. And she's saying as a clergy person, you're not going to feel shy. Why? Because you know that you have the knowledge. So these are examples of what people say who have actually gone through the workshop. And so in Zambia, as well as many other places, you're either infected with HIV, or if you're not, you are affected because it affects your community. On the second day, remember I told you after they get the information and go home and sleep on it. And one of the funny things is we know we've been effective when they come back the second day and somebody says, well, Professor Fuller, if we can get malaria from a mosquito, why can't we get HIV from a mosquito, right? And so I know when I hear that question that they got it. And so these are small groups that are strategizing using the knowledge they just got the day before with what they know about their community as to how to use that information. And our job at this time is done. And then the last couple of slides here, religious leaders then act. They engage their community confidently and willingly. And these are a couple of quotes. I'll just read one of them. A clergy person expresses the new perspective. We can act differently from what we used to act. If we are to change the situation with HIV and AIDS, then we should work as a team, both in the church and with medical personnel. And so what he's saying is that we need to have trusted messengers who can give us the knowledge, and with our own value knowledge, we can change this scenario. And so they do. They go out and open schools for vulnerable children. They form support groups for women who are dealing with HIV, who've been kicked out of their home and accused of sleeping around because now they brought this to their family. This is a home care team that was trained by a Ministry of Health organization after the pastor realized that, yes, I do want people in my community to be the go-between the clinics and the people who are home. So these are some of the things that we do when we go back for the site visits to see what the clergy who went through our workshops do with that information. So in summary, Dr. Spriggs, if you're still there, networks, and now we're not talking about knocking on doors, we're talking about using the highest authority of a network, and it could be any network, it doesn't have to be religious leaders, are a low-cost, highly effective way to move science advances to implementation. The in-depth science focus decreases fear and mobilization. People are fascinated to understand how these things work. The informed, influential leaders then model things like HIV testing, and they're willing to talk about things that before were taboo or things that would not be mentioned because they didn't want to be caught not knowing about it. And then participants appreciate the insight gained and share that with their family, friends, and community. So in conclusion, equipping religious leaders or whatever the network is as trusted messengers means to combine the power of science advances with access and influence of a trusted network of leaders, somebody in the community who has credibility to do effective partnerships towards, in my case, HIV/AIDS or COVID-19 or whatever the need may be. This opens a gateway to help communities navigate those complex social, behavior, and economical issues that we can't fix. We can't fix the gap in income. We can't fix many of the lack of transportation or the areas we live in, but we can help people understand that they can do certain things that will prevent these infectious microbes. So the trusted messenger idea is religious leaders can help to reframe community perspectives, perceptions about disease control and prevention. And that's what we've been doing so much with COVID this year. Pandemics have global presence, but so do trusted messengers, whether they're scientists or others. And so this just is pictures from Zambia. Twatutela means thank you in one of the languages. We believe this intervention can work if it's utilized with some of the nuances in urban or rural areas. And then these are acknowledgements of all the people, both the AME Church and the bishops and SEDA, many students who I've worked with, my public health colleagues, the University of Michigan funding systems, the COVID-19 partnerships, including the International Health Commission of the AME Church, and then some of our funding sources. So the last slide is just resources. And so that's what I wanted to share about how we need to engage with community. And there are ways to do that when you allow basic scientists to be basic scientists, public health scientists to do that. And then you have a few people who are able to combine or utilize both areas to get to people what they need to know to use the science that we've been fortunate enough to be able to do. So thank you for your attention.
Thank you for that amazing presentation. I am grateful for our digital platform so that people from all over the globe can hear your presentation. But I'm mad because I feel like if we were in person, you would be getting a standing ovation right now. So
Aw.
my claps will have to do. Thank you so much. That was a really important message, especially for Community Day for Black and Micro. I have a question to start with, talking about getting scientists out and being trusted messengers. Uh, with the training that we get right now, a lot of emphasis is put on doing the basic science, getting grants to fund the basic science, and it's just kind of a cycle in that way. What do you think that training program should be doing to change and equip scientists to better have the skills that they need to get out into the community and share this science? Because it doesn't really help if we know how the vaccines work and we know how COVID works, but obviously it's not translating. And so how can training programs help scientists that they're raising up for the next generation be better at this?
Yeah, I hear the question and I know the dilemma, and I have to say I don't know the answer. I can only give you one example with this process. And by the way, this is not easy and I don't recommend it for everyone. Let me just say that there's such a need for scientists in the basic science labs, in the clinical labs, to do what we do well. I mean, I didn't ask for this, I kind of stumbled into it. And as I say, I picked up the breadcrumbs along the way for the next step. There are not many ministers who are also scientists or virologists who are also ministers. But so what you do, what you are trained to do and what you love is what you should do. But in terms of answering your question, I think that a course that we developed called The Global Impact of Microbes actually takes students, either undergrads or it could be for grad students, into a real cultural immersion where they realize that the knowledge we have is just a small piece of the puzzle, but learning the customs of a place and respecting that, or knowing why certain things are done a certain way. We do this exercise about how do you peel a banana. So let me ask you, Dr. Spriggs, how do you peel a banana? How do you peel a banana?
Crack the top and
Okay.
then peel it down on the side.
Okay, well, there are other people that peel it just the opposite way, right? And that works for them. That's just natural. And the whole point is one way is not better than the other way. They all make the goal happen of getting the peeling away from the banana so you can eat it, right? So having small ways that grad students and others learn to appreciate the cultural proficiency. I don't call it competency because I don't think we ever become competent. But what we learn is that there's so much we don't know, and we need to respect cultures, respect the way people do things, learn to work with people who speak different technical languages. So, you might be a microbiologist and someone's an engineer. Well, you don't see things the same way, but being willing to sit down and work through a challenge or a problem together and taking things from each other to apply to that problem can actually get us much further along. So I think helping students to realize, yes, you have to focus, but you also need a bigger perspective of what the expertise is that can be applied to the challenges. And maybe I'm studying on a little piece of it, and that's my piece, and that's good, and I'm going to do that very well. But respect that we need all the pieces at the table before we can make a difference. So I only know a few places in the country that are doing the cross-disciplinary PhDs. One of them is the Fralin Medical Institute at Virginia Tech that has a program that takes graduate students and also takes them through some community engagement as part of their training. This is not typical. And maybe it shouldn't be, because we need really good scientists who know how to do the grants, the publishing, the research to keep that going, to get the fundamentals. But I think we need a few people who can also do the cross-disciplinary work. And then we need to change our systems to value this sort of thing as well. That's something that I can't fix, but I guess I've kind of gone my own way to say I value more so knowing that I'm making a difference in the lives of people and students who go on to do amazing things, than that my university says, well, I value you so much, I'm going to do this, that, and the other for you. So for me, that trade-off has been worth it, but I don't want other people to have to make that trade-off. the systems in academia should value what we say we value. We say we do research, teaching, and service, and all those things should be valued in a way that's tangible. Yes, because research brings in dollars and dollars run the world, that's what's most valued. But teaching is not any less important, and the service where we actually get things to people so they can use it should be valued in real ways. And that's, I may never win that battle, but I put it out there because those of you who are coming after us hopefully will have an opportunity to continue to work away at that, because you're going to be the deans and the presidents and the chairs and the distinguished professors that can make policy changes. Universities are one of the slowest, as a matter of fact, what's been said is churches or religious organizations and academic organizations are the slowest to embrace change.
Yeah, thank you for saying that. A lot of Black and Micro members, or BMA members, the Black Microbiologists Association, are trainees, and just Black scientists in general, and underrepresented scientists. A lot of us have this like deep need to feel like we can give back to our societies, and we want to do the outreach, but we also want to do good science at the same time. And I think that when we try to do service, people often see this as like the opposite of doing good science, like you need to just focus on science, or it's like an extra, but it's not always valued. So what advice do you have for trainees, or no matter what stage you're at, who want to do good science and they're committed to that, but also need to be able to engage with their communities through service?
Okay, that's a great question, and I think the answer for me is really, to do good science, you have to focus on it. You have to be all in with that. There are phases, as my mom says, you can do everything, just not everything all at once, right? So when you're in the graduate training, really your goal is to get your degree, and that should be your focus. And of course, for some of us to be well, doing other things which help us to do better with our research are part of the process by which we are well. And so do some of those things, but don't let those things move you so far away from your goal that you're knocked off track, because the bigger impact will be later on, after the degree or after the position or wherever you want to go. The other piece is, I think, important, is to always know what's in your heart, right? You cannot not be who you are. And so if your community is calling, we know that lots of NIH data show that women of color and people of color are less likely to get grants funded, even if it's something that a person who is a white person or Caucasian person comes in with the same grant would likely be considered for a higher or a better score, because it would be considered a better idea. So we know there's bias in the NIH process. You remember the barbershop case where, taking, I'll just use COVID education through barbers and beauticians, because they're right in the community. When it comes from a person of color as a proposal, it's not taken serious. But when it comes from a majority person, it's like, oh, this is a brilliant idea, let's fund this. So those systems are things that we need to help unravel, but we can't unravel them if we're not in the system, if that makes sense. You have to be at the table in order to be in the conversation and to change what happens. So I would say, make sure you do what you need to do, and do it very well, so you can be at the table and you can be those chairs. You can have the authority and the position and the power to make the changes. But always take care of you. I mean, that's essential. So if it means, I need to take a day or a week or whatever and do something different, do that, because that's what's necessary. You have to be well.
That's great advice. I think, this has been awesome, but before we finish, I just want to ask, is there anything else that you want to emphasize to the Black and Micro community or allies who may be watching this presentation, and how they can support the Black and Micro community, or just final closing remarks?
So is there anything that I have said that I haven't said that I want to say? I think so. Thank you for asking. mainly that we're in the middle of a pandemic. We're in the middle of a historical time, and to be a scientist and particularly a microbiologist in a time like this is really amazing. We've seen people like Dr. Corbett making contributions from what she was already doing to others who are doing amazing things in this particular pandemic, to what you guys do with Black in microbiology. So I just want to encourage all of us to support one another and to realize that you're not in this by yourself. When I started graduate school, I love science. I was always curious, and I was like, why is that? How come that? Why does that work? But when I started graduate school, I didn't see people that looked like me as professors. And I was like, where are they? Surely I'm not the only one that loves this. And sure enough, I found them. They were in the research triangle in industry. But my continuing as a scientist has been in part because I realized that people that I love to do what I love to do, which is, I think viruses are amazing. And if I can learn as much as I can about them, that's like, and get paid to do that too, that's really cool. But other people want to do that. So that's what's kept me in academia, such that people can be what they see. It's hard to do that if you feel like you're all by yourself. So I just want to commend the Black and Micro, or the Black Microbiology Association for what you're doing, because this camaraderie and support is so essential to you guys being the best you can be, and realizing that contrary to popular belief, people of color have been tremendous scientists from back in the prehistoric time, is my guess, but they're documented cases. But the systemic systems of inequalities have tried to keep that a secret. And we need to make that known. You're going to do great, even greater things, and I'm really excited about it.
I'm not going to cry.
Me either.
Thank you so much. This has been amazing. And you speaking on Community Day means so much to us. And the Black and Micro community very much appreciates you staying in academia and being just a light for us to see that we can also become scientists and professors. So thank you for your presentation today and for talking with us. And thank you for just your life and being here and being a part of our scientific community.
It's a pleasure. I appreciate the invitation. And this is not for everybody. So let me encourage you to stay your course and pick up the breadcrumb that's in front of you next to know what works for you. What I do may not work for you, but you will find what works for you. And I'm excited to watch it happen.
Thank you. Thank you so much. So tune in for our panel tonight at 6 p.m. And thank you, Dr. Fuller, once more for recording this talk with us so that everyone can watch it. Thank you.